Thank you for watching our mini lecture on enzyme kinetics and inhibition. We are going to start off by talking about enzymes. So this is an enzyme. And in the human body, there are tens of thousands of enzymes that aid in chemical reactions within the cells. Enzymes provide so many biological functions that they are actually an essential part of how we live. So it's important to know what an enzyme is. An enzyme is a type of biological molecule that acts as a catalyst to help with performing complex chemical reactions in our body. So a catalyst is a substance which speeds up a reaction, but remains chemically unchanged at the end of the reaction. It does not serve as a reactant or a product, but rather provides another process by which the reaction can occur. This process has a lower activation energy, which ends up increasing the rate of reaction. So activation energy is defined to be the minimum amount of energy required for the reaction to occur. And we see this in this graph. We see the red line is the process without an enzyme, and the blue line is the process with the enzyme. Both processes have the same reactants and the same products. They even have the same overall energy released during the reaction. The only thing that's different is the amount of activation energy required. So the process with the enzyme has a lower activation energy than the process without the enzyme. So the process with the enzyme increases the rate of reaction. So we talked about the functions of enzymes. One function in particular is when we eat. When we eat a piece of meat, a type of enzyme called a protease would go to work to help break down the bonds between the amino acids. And speaking of amino acids, enzymes are actually composed of amino acids and therefore are considered to be proteins themselves. However, something should be clarified before we continue. With the exception of ribozymes, all enzymes are proteins, but not all proteins are enzymes. Similarly, all enzymes are catalysts, but not all catalysts are enzymes. This is similar to saying that all sharks are fish, but not all fish are sharks. And we're glad to make that distinction. So this leads us to the enzyme substrate reaction. So this is the equation showing the different steps of the reaction, and below is a visual representation of what is occurring. So enzymes can only be used for a specific function. They are highly specific and they will only bind to one type of substrate. So this is shown by the shapes of the active site of the substrate. So it has like this weird U, V looking shape, and so does the substrate. The enzyme is the gray sort of circular thing, and the substrate is the green thing. So when they combine, the enzyme changes shape slightly as the substrate binds, and they form together an enzyme substrate complex. While they are in the enzyme substrate complex, the substrate becomes the product, and the complex becomes the enzyme products complex. Finally, the enzyme releases the products and resumes its role as the open enzyme, and it waits for another substrate to perform the function again. So this is the active site right here. The active site is a region on the enzyme where the substrate molecules bind. It consists of residues that form temporary bonds with the substrate and residues that catalyze a reaction of the substrate. So we see that the blue is where the substrate would bind, and the red is where this catalytic site is. The active site is usually a groove or a pocket on the enzyme, which can be located in a deep tunnel within the enzyme. So going back to the previous slide, when the substrate binds to the enzyme, we said it forms the enzyme substrate complex. While a part of the structure, the substrate is transformed into the product, and the enzyme product complex releases the product, and the enzyme is free to perform its function again. Notice that the enzyme remains unchanged at the beginning and end of the process, but the substrate is transformed into the product. So this is really important to pay attention to. So we have studied kinetics in this class before, and we all know that kinetics is the study of the rates of chemical reactions. Therefore, enzyme kinetics is the study of the rate of reactions that are catalyzed by enzymes. The rate at which an enzyme works is influenced by several factors, such as substrate concentration, temperature, pH, and the presence of inhibitors. The substrate concentration factor can be explained in terms of collision theory, which we also talked about earlier in this class. The more substrate molecules available, the quicker the enzyme molecules will collide with each other and bind. As the amount of substrate is increased, so is the rate of reaction. However, there however, as the substrate increases a lot, there are no more active sites for the substrate to bind to, and very high substrate concentrations of the very high substrate concentrations overwhelm the enzyme. And at this point, increasing the amount of substrate no longer affects the rate because the enzyme has become saturated. The point of saturation is when maximum velocity has been reached. Velocity then becomes independent of the amount of substrate. So when we talk about velocity, we're talking about rate, and that's in molarity per second. And we're talking about concentration of substrate, we're talking about molarity as well. So like the molarity of the substrate. So we see this line at the top right here with the arrow. That is the maximum velocity. And also notice that the plot of rate versus the concentration of substrate is not linear. And that is because of this relationship and the saturation point. So temperature also has an effect on rate, and this can also be explained in terms of collision theory within a certain temperature range. Molecular motion and, obviously, hence collisions between enzyme and substrate speed up as the temperature rises. But as enzymes are protein, there is an upper limit beyond which the enzyme becomes denatured and ineffective. And we see that in this graph. A real application of this is when we are ill with the flu. We often experience a fever. Our bodies increase in temperature in order to denature the infecting enzymes that are causing the illness. However, we are not able to function at this temperature for a long period of time because it would also begin to denature our own enzymes that keep us functional and living. So the pH also has an effect on rate of reaction. pH can alter protein conformation because certain amino acids become protonated or deprotonated at different pH. The effect is more severe if the enzyme's active site actually contains these types of residues. So inhibitors are substances that reduce the rate of reaction of enzymes. There are two broad categories: irreversible and reversible. Irreversible inhibitors cannot be unbound from the enzyme, but reversible inhibitors can, hence the name. So in this picture, we see the reaction occurring without any inhibitor. So this is like that enzyme substrate reaction we were looking at before. So we have the active site, the substrate binds, it forms the enzyme substrate complex, the product is made and released. When an inhibitor is involved, it also typically binds to an active site, which then blocks the substrate from being able to bind. Although in many cases inhibitors bind in the same binding site as the substrate, which is the active site, this same site binding is not a requirement. An inhibitor can also bind to what is called an allosteric site of the free enzyme and prevent substrate binding as long as it does not bind to the allosteric site when the substrate is bound. So it can only so the substrate here, if this was bound and it formed the enzyme substrate complex, this inhibitor could not bind in this allosteric area. Allosteric sites allow effectors to bind to the protein, often resulting in a conformation change involving protein dynamics. Allosteric sites are physically distinct from the active site, and this is portrayed by the allosteric site being sort of like square shape, and then the active site being kind of triangular shape. And that sort of conformation or physical change is shown when this inhibitor lays over the active site, which prevents the substrate from being able to bind. Although there are many types of inhibition, the type of inhibition we will be focusing on is competitive inhibition, which is reversible. A competitive inhibition is a form of enzyme inhibition where binding of the inhibitor to the active site on the enzyme prevents binding of the substrate and vice versa. So we saw that in the image before. In competitive inhibition, at any given moment, the enzyme may be bound to the inhibitor, the substrate, or neither, but it cannot be bound by both at the same time. And we see that here. So here is the inhibitor, and it is bound, and therefore the substrate cannot be bound. But when the substrate is bound, the inhibitor can't be bound. A competitive inhibitor has a higher probability to bind to an enzyme when the substrate concentration is low than when the substrate concentration is high, because they compete for the enzyme. If the enzyme inhibitor binds to the enzyme, it can be reversed by increasing the concentration of substrate. If the concentration of substrate is high enough, the enzyme inhibitor complex will return back to the inhibitor separate from the enzyme. At this time, the enzyme and substrate have an opportunity to bind and form the enzyme substrate complex. So this is a graph of the effects of an inhibitor, a competitive inhibitor versus the enzyme reaction without an inhibitor. So we saw this graph before. The green line represents the enzyme substrate interactions without an inhibitor. And it's got that nice curve, and then it reaches its maximum rate or velocity. Then we see with the competitive inhibitor, it takes a lot longer for it to reach that saturated part. So a competitive inhibitor slows down the rate of reaction. So the activity you will be doing in class will be modeling enzyme substrate interactions. The focus will be how different concentrations of substrate and inhibitor will affect the amount of product collected. So be sure to pay attention to the key features we covered in this video, and we will see you on Wednesday.